Hello everyone and welcome to today's webinar, Beyond Infection Prevention and Control, How Genome Sequencing of AMR Pathogens Can Improve Antibiotic Stewardship and Patient Care. I am Kaylee Bach of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by OpGen. To learn more, visit opgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Arne Materna, CEO of ARES-Genetics, an OpGen Group company. Dr. Materna, you may now begin your presentation.
Hello everybody, and welcome to this latest installment of our webinar series on antimicrobial resistance. My name is Arne Materna. I am the CEO of ARES-Genetics, and over the course of the webinar we will explore the positive impact that whole genome sequencing can have on antibiotic stewardship in general and on patient care and healthcare economics in particular. As always, first a few words about ARES-Genetics. During previous webinars, we have already explored the root causes for antimicrobial resistance, the need for antibiotic stewardship to counter the spread of AMR, and the positive effect that pathogen genome sequencing and surveillance can have on antibiotic stewardship efforts. With the ECCMID event just behind us, we will start today with an update on the latest numbers quantifying the global burden of AMR. After briefly revisiting the benefits of genomic surveillance and pathogen whole genome sequencing, here abbreviated with WGS, we will dive into recent advances in accuracy and turnaround time of whole genome sequencing and the use of machine learning for the prediction of antibiotic treatment response directly from bacterial genome data. I will conclude the seminar by reviewing studies that explore the clinical utility of whole genome sequencing and present data on healthcare economic and patient benefits. Let's begin with an update on the global burden of AMR. A study dubbed the GRAM study, GRAM thereby standing for the Global Research on Antimicrobial Resistance Project, has found much attention. The study was published in The Lancet this year, and it is offering a comprehensive analysis of the global burden of AMR. It shows that a staggering 4.95 million deaths in 2019 alone were associated with bacterial AMR. Associated with thereby means that if all drug-resistant infections were replaced by no infection, then 4.95 million deaths could have been prevented in 2019. This number has put bacterial AMR on place 3 of the leading underlying causes of death, only behind ischemic heart disease and stroke. A more conservative 1.27 million deaths were directly attributable to AMR. Directly attributable, thereby meaning that if all drug-resistant infections were replaced by drug-susceptible infections, then 1.27 million deaths could have been prevented in 2019. In other words, 1.27 million deaths were caused by the effect of AMR on infectious diseases. If we compare this number to the 700,000 deaths due to AMR in 2014, this number is an estimate in the O'Neill report published in 2015, then the fatalities due to AMR have grown by more than 81% in only 5 years. These numbers emphasize the pandemic nature of AMR, which only underscores the importance of urgent and globally coordinated action. The GRAM study further revealed that the AMR all-age death rates are highest in low to mid-income countries in sub-Saharan Africa, South Asia, Eastern Europe, and South Latin America. This makes AMR not only a major global health problem, but a particularly serious problem for some of the poorest countries in the world. As you can see in this chart, all 6 of the leading pathogens contributing to the burden of AMR in 2019 matched the ones previously identified as priority pathogens by the WHO. They include in this order Escherichia coli, Staphylococcus aureus, Klebsiella pneumoniae, Streptococcus pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. In the high-income superregion, approximately half of the fatal AMR burden was actually linked to the two pathogens Staph aureus, with 26.1% of deaths attributable to AMR, and E. coli, with 23.4% of deaths attributable to AMR. By contrast, in sub-Saharan Africa and other low-income regions, the leading pathogens were distinct from those of the high-income superregion, and each represented a smaller share of the AMR burden, complicating the issue in those countries. The leading types of infections contributing to the AMR burden globally include lower respiratory tract infections, bloodstream infections, intra-abdominal infections, urinary tract infections, and TB. This figure explores the antibiotic treatment options that were critically impacted in their efficacy by AMR. The analysis showed that resistance to fluoroquinolones and beta-lactam antibiotics accounted for more than 70% of deaths attributable to AMR across pathogens. Beta-lactam antibiotics thereby include carbapenems, cephalosporins, including third and fourth generation cephalosporins, and penicillins. Critically thereby is that these affected antibiotics are often used as a first line for empirical therapy of severe infections. In 2017, the WHO published a priority list of pathogens to guide the development of new and effective antibiotic treatments. Interestingly, only 5 of the 7 top pathogen drug combinations that the Graham study estimated to have caused the most deaths in 2019 are currently on this WHO priority list. For instance, fluoroquinolone-resistant E. coli are not included, and methicillin-resistant Staph aureus, while the pathogen drug combination causing most deaths in 2019, is listed as having a high but not a critical priority. Findings like the ones presented by the authors of the Graham study clearly show that surveillance data are crucial to inform policies. Given the rapid growth of AMR and that we review data from 2019 in 2022, shows that there is an urgent need for more continuous and globally integrated surveillance. The authors of the Graham study therefore concluded with a set of noteworthy key insights. First and foremost, the world is simply not doing enough to address AMR. Our current action plans are not ambitious or fast enough to control the threat. These estimates are a warning signal. Common infections have already become harder to treat, putting extra pressure on frontline healthcare workers. Immediate actions are required to help countries around the world protect their health systems against the threat of AMR. The authors also make policy recommendations, for instance on surveillance. They urge greater action to monitor and control infections globally, nationally, and also within individual hospitals. This requires accelerated support for infection prevention and control, as well as for antibiotic stewardship measures to reduce the use and overuse of antibiotics. Especially antibiotic use unrelated to treating human disease has to be reduced, but also antimicrobial treatment of infections has to expand access to life-saving antibiotics where needed and effective, while minimizing use where they are not effective or necessary. The use of effective antibiotics while avoiding overuse of ineffective or unnecessary ones is in a nutshell the definition of antibiotic stewardship. The question, of course, is how to identify treatment options that will not be met with resistance in a given outbreak or patient. The WHO, in its Global Action Plan on AMR, emphasizes again that the optimal antibiotic use reduces the spread of AMR and improves patient outcomes. The action plan proposes to improve antibiotic stewardship by focusing on technical solutions that support, on one hand, the rapid diagnosis of resistance, as such diagnostics can help inform a more targeted antimicrobial therapy. On the other hand, the majority of antibiotics are still empirically prescribed, informed based on surveillance data. Therefore, solutions for surveillance are needed that not only track the emergence and transmission of pathogen outbreaks, but also of the pathogen's ability to meet different antibiotic treatments with resistance. Let's take a look at the technologies that are in the clinic today. The standard of care in infectious disease diagnosis are bacterial cultures, which are routinely used for the identification of infective agents and the determination of suitable antibiotic treatment options. When a culture comes back positive, meaning pathogen growth is present, then antibiotic susceptibility testing, also referred to as AST, is performed to determine which antibiotics are effective and which will likely be met with resistance. While these culture-based methods in AST are being considered the gold standard, they are often labor-intensive and may miss difficult-to-culture pathogens. Also, turnaround times range from 1 to 3 days or even more, which is long considering that patients with acute infections require urgent care within hours. This lack of sensitivity and speed is addressed by modern molecular tests relying on PCR. Leading PCR panels, such as the tests available on the Curatis Univero system, achieve very high sensitivities and can return results in a matter of hours, even pushing the limit below 1 hour. These powerful systems can screen for up to a few hundred biomarkers in a single test, enabling them to simultaneously screen for pathogen and AMR markers. At OpGen and at ARESgenetics, we aim to complement such rapid molecular tests with sequencing technology. Given that today the number of known genetic markers for AMR is measured in the thousands, sequencing technologies hold the promise to simultaneously and comprehensively screen for any pathogen and all known genetic markers for antibiotic resistance, while at the same time delivering highly resolved information on outbreaks and on transmission patterns. Delivering on this promise has firmly emerged as a focus for ARESgenetics. Our goal is to develop sequencing assays in bioinformatics that identify pathogens and AMR markers, that offer genotype and outbreak cluster information, while also emulating the advantages of culture by predicting whether detected pathogens are susceptible or resistant to antibiotic therapy. Pathogen whole genome sequencing, or here again abbreviated with WGS, is increasingly adopted by clinical labs, and its utility for microbial typing and epidemiology is already undisputed. It is the first technology that, thanks to an enormous sampling depth, is not only very sensitive but also captures the entire genome, allowing for an exhaustive characterization of genetic AMR markers in a single test. And as I mentioned, comprehensive genomic information generated by WGS provides us a chance to use artificial intelligence and predictive modeling to emulate culture-based AST and predict which antibiotics represent suitable treatment options. With sequencing costs and turnaround times rapidly decreasing, the opportunity for complementing molecular testing, thereby revolutionizing the standard of care in clinical microbiology, is now no longer out of reach. The need for whole genome sequencing is in fact pervasive. In January, July, and again October 21, the International Health Regulations Emergency Committee for COVID-19 has recommended to increase global sequencing capacities. The IHR encouraged the rapid sharing of data, including metadata, and has asked the WHO to actively support countries to strengthen their systematic genomic surveillance. Between March 21 and January 22, there was in fact a 14% increase in the proportion of WHO member states with sequencing capability for SARS-CoV-2. It was deployed in over 2/3 of the member states in total. This is good news, but it means that there is now the need to sustain the gains made and strengthen WGS capacities further. In response, the WHO has just published a 10-year strategy for genomic surveillance. The goals of the strategy are to strengthen genomic surveillance for pathogens with pandemic and epidemic potential, including for AMR pathogens, and to scale genomic surveillance for quality and for timely and appropriate public health actions within local to global surveillance systems. Now let's revisit how WGS can assist us in the fight against AMR. Here a quote by Louisa Gordon, David Patterson, Patrick Harris and co-workers, the authors of a study that we'll take a closer look at in a minute. The authors state that when compared to the standard of care, whole genome sequencing can confirm or refute suspected related cases in infectious pathogens. In other words, it can detect outbreak clusters and provide an early warning. It can discriminate between different strains, which in other words allows for better patient management by the infection prevention and control teams. And finally, whole genome sequencing can classify novel pathogens, which in turn improves the diagnostic yield potentially leading to more detected and appropriately treated infections. Given the potential value inherent to whole genome sequencing, the question is, how can we speed up its adoption, especially given that the workflows for sample processing and data analysis can be quite complex and sometimes laborious. Clinicians tell us that the answer to this question has multiple facets. For one, it is important to demonstrate sufficient diagnostic performance of WGS. Yet, in addition to being accurate and reproducible, a broader adoption of whole genome sequencing will also require an understanding of its clinical utility. In other words, its proven capacity for improving patient outcomes, while ideally reducing the total cost of care. Over the following slides, we will therefore explore both aspects of the clinical utility of whole genome sequencing, and we'll start with taking a look at its diagnostic performance. To ensure the accurate performance of our platform for isolate sequencing at ARES, which we have dubbed ARESiss, and which we have made available as a service last year, we routinely validate our workflows and publish the results along with validation protocols. Here, the example of a study we've published in 2020, in which the team has validated ARESiss and the data analysis following CLIA guidelines. CLIA is a US standard for laboratory developed tests. The performance was assessed using well-characterized reference materials and a number of parameters, including accuracy, repeatability and reproducibility, as well as sensitivity and specificity were recorded. The study showed that the ARES process for isolate sequencing 100% accurately and reproducibly identified and genotyped pathogens. Also, AMR marker detection was highly accurate with sensitivities and specificities exceeding 95 and 99% respectively. With nanopore sequencing becoming increasingly available, we have decided to conduct a second multi-center validation of the workflow for isolate sequencing, ARESiss, this time using the ONT platform. Nanopore sequencing is an interesting platform for clinical microbiology for a number of reasons. The turnaround time is very low, theoretically enabling same-day reporting and workflows as short as 6 hours. The technology also generates long reads, which offer interesting advantages for the assembly of bacterial genomes and, for example, for the recovery of plasmids. Finally, the small footprint and the low capital equipment cost, as well as per base cost, make this an interesting platform for clinical or surveillance applications, also in low to mid-income countries. Nanopore sequencing works by translocating a single DNA strand across a biological pore and a membrane. A steady flow of ions through that pore creates a current which can be measured and which fluctuates in a DNA sequence-specific pattern as the DNA is translocated through the pore. The downside is that both the nanopore itself as well as the DNA translocating effect rely on live biological proteins, which introduce biological variability into the system, leading to a, in theory, less accurate base calling compared to, for example, Illumina short read sequencing. For our study, the well-characterized set of reference materials was subjected to DNA extraction by different vendors, and the resulting DNA extracts were aliquoted and sent to Oxford Nanopore Technology-enabled partner labs for library preparation and sequencing using the 10.4 flow cells and the rapid library prep chemistry by ONT. The results of this study have been summarized in a white paper, which you can find on our website. A manuscript with a full assessment of the diagnostic performance of our workflow and our bioinformatics in the hands of researchers at Johns Hopkins Hospital has been submitted. Indeed, the base calling quality was reduced. We obtained quality or Q-score on the Phred scale of about 11 to 12, compared to much higher Q-scores of 30 that we typically see with Illumina data. However, the assembly process resolves some of the associated problems. After de novo genome assembly, we assess the average nucleotide identity of assembled genomes with a known public reference and determined that the error rate was only 3 to 4-fold higher compared to Illumina-based assemblies. Despite this unresolved difference in sequencing error rate, the ARES bioinformatics pipeline for whole genome sequencing on the ONT platform performed well. The accuracy and reproducibility for pathogen identification was 100%. We also obtained high accuracies for AMR marker profiling and plasmid detection, which were only slightly below the performance of Illumina data. Most satisfying was the fact that the antibiogram prediction directly from genomic data using our predictive antibiotic susceptibility testing pipeline performed very well with an accuracy of up to 97% and in line with Illumina data. The only area where the nanopore technology needs to improve further is bacterial typing using core genome MLSD, as the involved bioinformatics are more sensitive to sequencing error. We have enrolled with ONT in the so-called Q20 program, the Q20 chemistry early access program, which is promising drastic quality increases. The first test runs using the new chemistry, also for core genome MLSD typing, look promising, but further testing is required at this point. We've now established that the diagnostic performance of WGS is high, offering results that are both accurate and reproducible. In order to maximize the impact of a technology on AMR outbreaks, however, we established that a useful diagnostic has to deliver insights into antibiotic susceptibility and resistance for a given outbreak or for an individual case. I will therefore review over the next couple of slides a machine learning assisted process that we at ARES-genetics refer to as predictive susceptibility testing, or PASD, which is the prediction of antibiograms directly from genomic data. To achieve antibiogram prediction, we use artificial intelligence, which is trained on data we continuously aggregated in a proprietary database we call ARESdb. ARESdb holds, to the best of our knowledge, a leading collection of genome and associated phenotypic resistance data from clinical isolates. Currently, the models to predict antibiotic susceptibility are trained as stacked ensemble models, which associate phenotypic AST results with a variety of different genetic markers, including k-mers or gene or protein annotations. This model-based approach can be complemented by a rule-based approach, but it can, other than purely rule-based methods, take into account the synergistic or antagonistic interaction of multiple AMR genes or AMR-associated mutations and the interplay between multiple antibiotic compounds. As the figure in the middle illustrates, we not only rely on the presence or absence of certain resistance genes, but we also consider changes in non-coding regions of the genome, as well as novel markers that our predictive algorithms have discovered. We demonstrated in several studies the high categorical agreement between standard of care results with the resistance phenotypes we predicted based on WGS data. These studies include, for instance, the first multi-center evaluation successfully demonstrating the feasibility and performance of predictive AST for 13 pathogens causing lower respiratory tract infections. Some of the study results are shown here. Allow me to walk you through the somewhat data-dense slide. The matrix on the right-hand side shows along the horizontal axis the samples for the various pathogens reported by the standard of care. The vertical axis lists the different antibiotic compounds by compound class that were tested for. Each intersection in this matrix represents a pathogen-drug combination and requires a separate predictive model to be trained. The numbers for each of these intersections, or the fields in this table, show the categorical agreement in % between predictive AST and culture. If you exclude all pathogen-drug combinations for which pathogens are intrinsically resistant, highlighted here in blue, and those combinations for which the standard of care method did not detect any resistance, highlighted in gray, then the remaining white fields of the table are the areas in which a direct comparison of ARES predictive AST and culture-based AST is possible. As the green fields indicate, we were able to demonstrate high concordance, reaching for most of the pathogen-drug combinations values significantly above the regulatory threshold for AST devices of greater than 90%, often achieving up to 100%. Studies like this are not only important for clinical validation purposes, We also embrace them as they give our data science teams clear indicators on which pathogen drug combinations to focus our efforts on for predictive model development and for database curation. This slide shows the results of such an effort to improve model performance for a given drug-bug combination through focused biocuration and model training. Together with our partners, which include, for instance, reference labs, we generate and aggregate isolate genomes and the corresponding phenotypic data on AMR. The data show that by selectively increasing the number of data sets from isolates with specific phenotypes, we can systematically improve categorical agreement as well as errors, shown here by the decrease of the so-called very major error rate, which is the false susceptible rate. In summary, biocuration and model training go hand in hand and can improve model performance for each bug-drug combination to levels in line with IVD device regulation. The benefit for our partner thereby resides in being able to accurately predict treatment response directly from genomic surveillance data, and of course in having access to an up-to-date AMR marker database. This slide intends to show the impact that biocuration has on average on the performance of our predictive AST models. The figure on the right shows percent categorical agreement between prediction and the standard of care at the top, and the error rates for very major error, and minor errors at the bottom. On the very right, you can see the average performance over all predictive AST models we evaluated in the original multi-center study. Again, these values are averages, also including models which in our 2020 study have underperformed due to insufficient training data sets in the ARES database. Those models correspond to the white fields in the matrix I just showed. The middle of the chart on this slide shows the results of 3 independent studies performed using our 2020 predictive models on different subsets of the full spectrum of drug-bug combinations. Finally, on the right-hand side, highlighted in green, you can see the average performance across our predictive AST models when trained based on a more recent version of the ARESdb. Categorical agreement is very high with an average 96%, and also very major errors and major error rates have significantly improved. To ensure that predicting resistance and susceptibility is not only accurate, but also accessible to researchers and clinicians without expertise in bioinformatics or AI, we have launched last year ARES-cloud, a user-friendly web application which fully automates predictive AST, in other words, the antibiogram prediction, directly from genomic data. The platform generates reports which, in addition to antibiograms, also include information on sequence quality and contamination, of course on pathogen identity and core genome MLSD genotype. To round out the solution, we have enabled users to drill into rich information on individually detected AMR markers, their prevalence, and their prognostic performance. Also included is information on plasmids and virulence factors. All reports, some examples of which are shown on this slide, can be explored online or exported with a click of a button. For convenience, ARES-cloud also includes a set of comparative analysis tools for the investigation of local bacterial outbreaks. In summary, we have now established that whole genome sequencing offers accurate and reproducible information on pathogen identity, genotype, and outbreak clustering. WGS offers insight into virulence and AMR, revealing genetic markers, mobile elements, and plasmids. WGS data in conjunction with artificial intelligence can be used to accurately detect AMR and predict susceptibility, thereby offering a route to improve treatment guidance and antibiotic stewardship. The new long-read platforms are becoming a viable option for enabling same-day reporting and to lower capital equipment cost, which makes whole genome sequencing increasingly suitable for the clinic and for surveillance in low to mid-income countries. And finally, we have shown that bioinformatics can be fully automated and delivered through secure and user-friendly applications. But not only has there been tremendous progress on the side of diagnostic performance, also the question about the clinical utility of whole genome sequencing is being answered. As a quick reminder, clinical utility is defined as patient outcome divided by the total cost of care. This means to fully describe the utility of any new intervention, its impact on both outcome and cost has to be understood. And a number of researchers have focused their efforts on furthering our understanding of the clinical utility of WGS. Among the early adopters of clinical whole genome sequencing were the German team of Alexander Mellmann, Doug Harmsen, and co-workers. In a 2016 study, this team assessed the overall sequencing costs using a case during which 550 isolates were obtained. Next, they calculated the additional direct healthcare costs plus the indirect costs resulting from blocked beds in the intensive care unit associated with cases that could have been prevented with whole genome sequencing. Interestingly, the direct and indirect healthcare costs were thereby very conservatively assessed. The team used cost data that at the time of publication were outdated by 15 years. I guess we can intuitively agree that within 15 years, costs for most things have gone up, while however the costs for sequencing in recent years have come down. Despite this conservative approach, the team calculated for this specific case involving again 550 isolates a total cost saving of 205,000 euros. As a positive side effect, the team also counted the avoided risks and detrimental health impact associated with patient isolation in an intensive care unit. More recent studies use statistical and mathematical modeling to estimate the cost effectiveness of routine whole genome sequencing as an infection control tool. The studies by Diamond and co-workers and Elliott and co-workers modeled the effects of whole genome sequencing on patient outcome and on healthcare cost based on data they acquired from AMR outbreaks with MRSA and OXA 181 E. coli respectively. Diamond and co-workers based their model on data generated during a 12-month prospective observational cohort study. They concluded that routine MRSA sequencing would result in fewer cases. They included both colonization and clinical infections, and to have a small but measurable positive impact on health-related quality of life on a per-patient basis. And that effect was regardless of whether these patients have been infected or were only tested for MRSA. The team also estimated that routine whole genome sequencing was likely to be cost-saving under the model assumptions. The team, including Elliott and co-workers, conducted a model simulation of the clinical utility of whole genome sequencing during an active outbreak in a hospital. Their model was motivated by and trained on data from an OXA 181 E. coli outbreak in April of 2017. The findings of their simulation reveal that whole genome sequencing of a potentially serious pathogen appears effective in containing an outbreak and minimizing hospital costs at the same time. They predicted that without the use of whole genome sequencing, the number of colonized patients would increase by 78.7% and the associated costs would increase by almost 67%. Finally, I'd like to highlight a study from 2021 conducted by Louisa Gordon, David Patterson, Patrick Harris, and co-workers. This is the team I quoted earlier. Motivated by the earlier promising findings on the benefits of clinical whole genome sequencing, this team decided to determine the effect on cost and patient outcome, but at a regional level. They collected data from 27 hospitals and considered the entire state of Queensland, which is the second largest and the third most populous in Australia, for their analysis. The costs were modeled based on costs for hospitalization, sequencing, and epidemiology. The assumptions on AMR incidence considered annual hospitalizations, which are 410,000 hospitalizations in this case, the point prevalence for healthcare-associated infections in Australia, which is 9.9%, and the multi-drug resistance rates and their trends. The costs were then aggregated for the entire state of Queensland based on the expected number of multi-drug resistant isolates arising in Queensland hospital patients given the point prevalence. The sequencing data from isolates for the study came from a 2-year research demonstration project of prospective whole genome sequencing for isolates of suspected outbreaks. The team compared the interventions whole genome sequencing versus the standard of care for multidrug resistant pathogens. It was thereby assumed that whole genome sequencing based surveillance would occur for the 6 common multidrug resistant organisms, for the 6 most common multidrug resistant organisms, including methicillin resistant Staphylococcus aureus, extended spectrum beta-lactamase producing Escherichia coli, vancomycin resistant Enterococcus faecium, ESBL producing Klebsiella pneumoniae, carbapenemase producing Enterobacterialis, and carbapenem resistant Acinetobacter baumannii. The study then compared the following primary and secondary outcomes for both interventions: expected hospital costs, counts of patient infections and colonizations, and deaths from bloodstream infections. The authors actually calculated costs annually across 5 years from the base year 2020 on. But for simplicity, I highlight the impact of only 1 year of whole genome sequencing. The study findings projected that in 2021, when only relying on standard of care testing, then 97,539 patients in Queensland were expected to be infected or colonized with one of the 6 multidrug resistant organisms. In contrast, a whole genome sequencing based surveillance strategy and earlier infection control measures could avoid 36,726 infected or colonized patients and avoid 650 deaths. These numbers, highlighted in green here on the right-hand table, deliver a very clear message about the patient benefit that can be realized through routine whole genome sequencing. Now let's take a look at the impact on healthcare costs next. The total cost under the standard of care was 170.8 million Australian dollars in 2021. Whole genome sequencing based surveillance added an additional cost of 26.8 million Australian dollars. This cost, however, was offset by fewer costs for cleaning, nursing, personal protective equipment, shorter hospital stays, and antimicrobials, leading to an overall cost savings of 30.9 million Australian dollars in 2021 alone. The authors also conducted a sensitivity analysis which showed that cost savings remained even when input values were varied at 95% confidence limits. While these studies rely largely on models rather than being fully retrospective in nature, the models rely on actual data from outbreaks, which had been managed using whole genome sequencing. Needless to say that these studies have implications for government resource allocation to healthcare. They also established a favorable value proposition for adopting pathogen whole genome sequencing into the routine clinical practice. I'd like to conclude this presentation with a short summary of what we have discussed. The Graham study impressively established that AMR, especially multidrug resistance in healthcare-associated infections, is on the rise, with fatalities due to AMR growing over 81% globally in only 5 years. As discussed also in previous webinars, antibiotic stewardship and the appropriate use of only effective antibiotics is a critical requirement to contain AMR. And antibiotic stewardship in turn depends on next generation diagnostics for AMR surveillance and for infection prevention and control. Whole genome sequencing is a promising technology proven during the COVID-19 pandemic and during countless bacterial outbreaks. Global health organizations urge healthcare systems to scale up and coordinate whole genome sequencing in the clinical routine. We've established today also, by reviewing a number of studies, that whole genome sequencing can be accurate and reproducible. Also, turnaround times and costs are quickly becoming conducive to routine testing in clinics and in low to mid-income countries. Finally, a number of studies on the healthcare economics of routine whole genome sequencing of AMR pathogens support the clinical utility both at the local and the regional level. With that, I'd like to invite you to get in touch if you're interested in whole genome sequencing and in improving antibiotic stewardship together. We'd be looking forward to meeting you either virtually or maybe in person at the ASM Microbe in June. Of course, this webinar series on combating AMR shall continue. We expect an invited speaker presenting at the end of June, so stay tuned. A big thank you to all our partners and collaborators, and of course to you for dialing in today and for your attention.
Thank you, Dr. Materna, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question here says, thank you for this interesting presentation. Susceptibility from genomic data is indeed the next frontier if we want to see the adoption of NGS in the clinic beyond surveillance. I assume that the predictive models work well in pathogens like Staph aureus or E. coli, for which susceptibility is linked to the absence of well-characterized resistance genes. Could your AI-based approach also help predict susceptibility for pathogens or antibiotics with more complex resistance mechanisms?
That's indeed a very good question. The short answer is yes. Common rule-based methods for the prediction of susceptibility work well for pathogen-drug combinations for which the presence-absence, for example, of acquired resistance genes are the dominant resistance mechanism. The prediction of phenotypic susceptibility becomes already much more challenging in cases where mutations in drug targets or in regulatory regions, for example, affect the AMR phenotype. So cases like carbapenemase negative Klebsiella have in fact motivated the approach that we are using. Wherever a number of different genomic features can either synergistically or antagonistically interact in their contribution to the AMR phenotype, this is where AI-based predictive models outperform the simpler rule-based methods, as we know them, for example, from ResFinder and other tools.
Great, thank you.
That addressed the question.
That was a wonderful answer, thank you. We have another question here. This one asks, we would like to sequence isolates to distinguish between outbreaks in our hospitals from community-acquired cases. The idea is to better inform the action of the IPC teams, but our shared NGS core cannot always return results fast enough. Sometimes we receive first data after more than 1 month, at which point they are useful for retrospective outbreak analysis, but not for decision-making in the IPC team. How fast can the ARESgenetics lab return isolate sequencing data, and can you help us detect outbreak clusters?
Yes. This is indeed a problem we also hear quite frequently in the community here in Europe. On one hand, clinical microbiologists try to find budgets for isolate sequencing, and on the other hand, data arrives so late that the IPC teams can essentially no longer act on them, other than maybe publishing a retrospective outbreak analysis. So to give timely input on whether a sample falls, in fact, into an outbreak cluster or is rather community acquired, we launched the ARESiss Express service. So ARESiss stands for isolate sequencing and is a sequencing service we offer. And the express version of this service converts isolates into clear reports. They include, for instance, information on genotype, but also clustering. And we return samples around in as little as 5 business days. For labs that in fact have in-house NGS capacity and could therefore accelerate this further, we have also made ARES-cloud, which is the software that our own lab uses for end-to-end data analysis and reporting. And we've made ARES-cloud commercially available as a web application as well.
Great, thank you. Our next question here asks, our hygiene department is collecting isolates to monitor the hospital environment for AMR pathogens. We would like to try metagenomic sequencing, but are concerned about sensitivity, especially for AMR detection. Do you have any advice on this?
Yeah, that's a good question. In theory, deep metagenomic sequencing can detect any AMR marker in a sample. But especially for, let's say, environmental surveillance, think of for example, of surface swabs or sink swabs, water samples, and so on. The bacterial DNA in a sample can be so sparse and the background of unrelated DNA so high that shotgun metagenomic sequencing is starting to struggle to achieve good sensitivity and reproducibility. And metagenomic surveillance essentially is a sampling problem. So the likelihood of sampling a pathogen or AMR marker when sequencing such a sample has to be shifted in favor of a robust detection. And we are doing this by developing so-called enrichment methods. For instance, ARES-UPA, which stands for Universal Pathogenome Assay, combines on one hand ARES-ID, which is our commercially available amplicon-based approach for the identification of pathogens, with on the other hand a hybridization capture approach. So this approach enriches for a panel of over 9,000 AMR markers, which we have selected from the ARESdb. So for any type of environmental surveillance in the healthcare environment or otherwise, I would certainly recommend to use an enrichment-based method like ARES-ID or the ARES-UPA I just mentioned. They're both commercially available, although I have to say that the ARES-UPA is currently only available as for early access testers, but we're certainly happy to help if you have a need there.
Great, thank you. Another question here asks, how can we watch the previous webinar series?
The previous webinar series are available on our website. You can find that under www.opgen.com/ares. And under the ARES brand page, you can find resources which include links to all the ARES webinars. But I think it's probably best if we just share the link with you in maybe all the attendees in a follow-up email, as this will also be the place where you can watch the webinar on demand and also find future webinars.
Great, thank you. All right, another question here. Do you have any idea why Mycobacterium abscessus is not included in leading AMR pathogen, even though they are highly resistant to most of the available antibiotics?
I see. So non-tuberculous mycobacteria like Mycobacterium abscessus, they certainly contribute more and more to healthcare-associated infections. But I think the reason it wasn't included in the study that the question is referring to is probably due to the fact that the study focused on respiratory tract infections. And as far as I know, does Mycobacterium abscessus rarely contribute to respiratory tract infections. It is much more common in soft tissue infections, skin infections, and so on. And I believe that might be the reason why it was not included.
Fantastic. Well, we are going to go ahead and wrap up with that final question there. I want to thank you again, Dr. Materna, for your time today and your important research. We would also like to thank LabRoots and our sponsor, OpGen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can also be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.